Welcome to Chapter 20, Antimicrobial Drugs. Okay, we'll talk about some terminology. We'll talk about a few tests to test the effectiveness of drugs. And then we will focus most of our attention on specific modes of action of different antimicrobial drugs. It's important that you memorize all of the specific drugs that I give you in this lecture. There are many more in your chapter that you are free to ignore. We will spend most of our attention on antibacterial drugs. We'll briefly talk about a few examples of antifungals, antivirals, and antiprotozoans. And then we will focus the rest of our attention on ways that microbes, and in particular bacteria, have come up with ways to get around these drugs, and we call that antibiotic resistance. However, we really should say antimicrobial drug resistance, because not everything is actually an antibiotic. And so that leads into our terminology talk. So chemotherapy is just using chemistry in the form of drugs to treat a disease. We are used to hearing chemotherapy, or for short chemo, when talking about cancer, but really anything that's targeted at treating a disease that you put into the body is chemotherapy. Antimicrobial drugs are specifically drugs to interfere with the growth of microbes, microbes that are in the host. This could be intracellular or not, and this could be bacterial, fungal, viral, protozoan, or even helminth. So this is the most general phrase that you could use is antimicrobial drug, and it's typically the most correct terminology to use. When we use the word antibiotic, we are actually talking specifically about a substance produced by a microbe to kill a different microbe. So a substance produced by a microbe that in small amounts shows inhibition or even killing of other types of microbes, okay? And then we isolate that and take it and use it for our advantage. Then all the other drugs are either modifications upon antibiotics, or they're completely synthetic. Selective toxicity is a key concept when thinking about drugs. You need them to kill the microbe or slow down the microbe without damaging the host. And you'll see some of the drugs we'll talk about, they don't have very good selective toxicity. They still have side effects, some quite dangerous for people, but it is the only, sometimes it's the only option, and the benefit of saving someone from a deadly infection would then be greater than the outcome of the side effects, right? So you always have to weigh the benefits from the side effects. So that's the therapeutic index, right? Risk versus benefit. Another thing that you need to think about is the drug target, right? So selective toxicity means targeting the microbe and not the host. So you want the actual target where the drug does its activity to be something that's unique to the microbe. If it's not found in the host, then typically the drug won't interact with the host and there won't be any side effects. That's not always true, because there can be other things involved, like the kidneys or the liver filtering the drug out, or some like secondary interactions somewhere in the body as well. But overall, that's the idea, is that if you find a unique target, something unique to the microbe, then that's the first step in the selective toxicity. Okay, so here is a brief example of some of the microorganisms that produce actual antibiotics. And you'll see that the vast majority of antibiotics are produced by Streptomyces or another member of the actinomycetes group. Recall that that this is the fungus-like bacteria, so it is bacteria. It's in that phyla Actinobacteria, and it's in the order of Actinomycetes. Okay, I do not expect you to memorize the entire chart. I do not expect you to memorize any of the species names. I just want you to remember Actinomycetes, and Streptomyces is within the group Actinomycetes, and they produce the majority of our antibiotics. Okay, you'll see though we do also have penicillin. You should also know the antibiotic penicillin comes from the fungi Penicillium. Okay, so those are the two most important things to know about this chart. Okay, so first we'll talk about some assays. You were introduced to disc diffusion already for chemicals. Now we're going to look at it for antibiotics. And then we'll talk about another method called broth dilution method, and a couple of key terms with that as well. So for each of these assays, disc diffusion, E-test, and broth dilutions, make sure you know the general flow, like what are you using and how are you using it. We can say things in two ways. Is the antibiotic successful at treating the infection, or you can say, is the bacteria susceptible to the drug? Okay, so the Kirby-Bauer method is this method of dividing a Petri plate which has a lawn of bacteria on it, and then placing these discs equal distance apart. And these discs have a diluted concentrations of drug, and typically you buy these discs already with the drug impregnated in them. So all you have to do is place the disc on the plate. The discs will be labeled with the concentration of the drug, and then you should also have literature to interpret the results. So this is the most widely used method to see for a particular type of bacteria which antibiotics would be a good choice, which ones will work. And I've said this before, but I'll say it again. Every time you go to the doctor's office and they prescribe you antibiotics, you should always ask for a culture to confirm that it is the correct antibiotic. And you should never take antibiotics if you don't actually have a bacterial infection. So most infections are actually viral, and antibiotics will not do anything to treat a viral infection. All right. So standardized amount of organism, right? So we know how much we're spreading across the plate, and it's uniformly spread across the plate to make that lawn. And then we place the disc with the drug on it, and we know the concentration of the drug. It will be on both on the bottle and on the disc itself. And then after you incubate, you look for that zone of clearance. As you see here, no zone of clearance. Here you have a nice zone of clearance, and you have to measure that in millimeters, and then you have to look at the standard table reference for what is considered susceptible. So if the zone of clearance is large enough, according to that reference table or chart that you look at, then you would say that the bacteria is sensitive or susceptible to the drug. Perhaps it's intermediate. It's not fully susceptible, but you did get some inhibition, and or it can be resistant. So we can say that, for example, this disc that doesn't have a zone at all, we know is the bacteria are resistant to that drug for sure, because there's no zone of clearance. But for these other ones, we don't know until we measure and compare it to the chart. So let's go ahead and watch this video on the disc diffusion method. You should pause my video, click on this link, and watch the disc diffusion method. You can watch the first part where they introduce the concept of the test. Then you can skip the middle if you want, and then carefully watch the results portion of how to interpret the results. Okay, so for the E-test, this is a slightly more advanced diffusion method where you're just looking at a single drug, and the drug is you'll see along the strip, you go from being the most concentrated to being more and more diluted. From right to left across the width of the strip, it's an even amount of the drug, but as you move down towards the bottom, you'll see the numbers which indicate less and less concentration, and so that will produce this nice kind of egg shape of a zone of clearance, zone of inhibition. Okay, and so this gives you a much better idea of what the minimum concentration of the drug is that will give you inhibition. Okay, so the MIC is minimal inhibitory concentration. That's the lowest concentration of the drug that inhibits growth of the bacteria. Again, you're working with a lawn of bacteria. Now in this plate it's red, so you're looking specifically at a plate where 5% sheep's blood has been added. So you're testing for something like one of those strep group A strep bacteria. And this is usually in micrograms per mil for the drug concentration. And I asked you what is the MIC of this antibiotic, but you can't really see these numbers very clearly. So hopefully you would just say, it's this, whatever number this is right here. And I think that says 0.94, but it's very tiny. Okay, so that's the E-test. And then broth dilution tests. Now, I'm not showing you any particular images here because there are quite a few different protocols, but the underlying concept is the same. So we'll just talk about the concept where you want to be able to determine both the minimal amount of drug to inhibit growth and the minimal amount of drug to actually kill the organism. So in BCs, the minimal bactericidal concentration to actually kill. So you're going to have different tubes of broth with the same amount of bacteria in each tube, and you will put different concentrations of the drug, and the first, the lowest concentration of the drug that inhibits bacterial growth, you would then do a subculture, and the subculture is going to tell you whether you killed the bacteria or you simply inhibited. So if the subculture doesn't grow any bacteria, then you know that you actually killed it. But if the subculture grows, then you know that you simply only inhibited. So that will differentiate between the MIC and the MBC. All right, so let's talk about specific drugs. So we have what we call a spectrum of activity, and this is this shows you like the group of organisms that the drug can treat infections for. So here we see a type of penicillin that is able to kill most gram-positive bacteria, not all, but most. Okay, You'll notice on this that the chlamydia and rickettsia are intracellular pathogens that are being grouped separately from the rest of the organisms here, and then also mycobacteria, which is also can be considered gram-positive, but it's really acid-fast, right? Myco from mycolic acid. It's being grouped separately as well, right? So we see a drug that's good at treating mycobacteria doesn't treat these other bacteria, right? We see streptomyosin, on the other hand, is a broad spectrum in that it's getting mycobacteria and gram-negative. Tetracycline, also a broad spectrum of a little bit of mycobacterium, gram-negative, gram-positive, and a little bit for chlamydia and rickettsia. However, with tetracycline, it's important that you know which generation, because there are different types, different generations that will be better at targeting specific groups. So in actuality, the specific tetracycline that you give, the specific generation of drug you give, is usually actually a narrow spectrum. It'll be really good at targeting something along the line that you see here. So you won't necessarily see this entire width for every single tetracycline drug, but somewhere along this line. Okay. And then we have the antifungals, anti-protozoans, helminths, and antivirals. And those, of course, are going to be relatively narrow spectrum. It's harder to make drugs for eukaryotic organisms because they're more similar to our own cells. There's fewer unique targets. Furthermore, viruses are here. They are not eukaryotes, they're not living, and they can be tricky to treat because they're using host machinery. They're taking over our cell, and so it can be hard to find targets that are not going to hurt us as well. Okay. Another reason why viruses can be tricky is that they have a high mutation rate. They're kind of sloppy with their replication of their genome, and so you can get very, very subtle differences within one person who's infected with a virus. They can actually have many different strains of the virus. That further complicates trying to treat a viral infection. Okay, so mechanisms of the mode of action of the antibiotics, and really this should say antimicrobial drugs or antibacterial drugs. Okay, so we could target cell wall synthesis, we could target protein synthesis, we can target the plasma membrane, and we can target the DNA replication or mRNA synthesis. And then we could also use competitive inhibitors of enzymes that are important for metabolism. Okay, so the first category we're going to look at are inhibitors of cell wall synthesis. And we're going to look at penicillin, amoxicillin, carbapenem, cephalosporin, and vancomycin. And so you see in this picture here that it's showing the cell wall. It's acting on the cell wall. All of these are going to kill the bacteria. And then we have another group. We have another group called inhibitors of protein synthesis. And so you see that the picture is pointing at translation, which is taking mRNA and making protein, so protein synthesis. We have a third group, which the picture shows the plasma membrane, so you're targeting the plasma membrane. The fourth group targeting DNA replication, And then lastly, the inhibitors of enzymes needed for the synthesis of essential metabolites. Okay, and so I've put the specific drugs that you need to know for each of these groups. So this is a very important slide for you to memorize. Okay, so inhibitors of cell wall synthesis. We're going to have two main modes of action in this group. We're going to have the beta-lactam ring antibiotics, and then we're going to have the polypeptide antibiotics. So I've given you some information for each of these, and then we'll look and look a little more closely at each of these.